Good morning once again, and today I will focus. The main point of my talk will be COVID-19 or SARS infection, actually, and the epigenetic mechanism that can play a role in its pathogenesis and then the late consequences in CNS, so central nervous system pathologies. But to start with, the first things first. So I will just guide you slowly through the field of epigenetics. Next slide, please. Next one. The next slide. Thank you. So epigenetic is a huge machinery that is located outside DNA sequence. And it has plenty of phases like non-coding RNAs, like chromatin remodeling, like histone modifications, and last but not least, DNA methylation. And we will focus on that today because that seems to be a very, very potent biomarker. So next slide, please. So 5-methylcytosine, so what we call DNA methylation, is the presence of 5-methylcytosine in DNA strand. It on a genetic level, it acts as cytosine. It makes a pair with guanine. However, on an epigenetic level, it is a very, very important switch about gene expression. Next slide, please. Okay, next one. So it functions as a switch. So if we have a lot of 5-methylcytosines, the gene expression is off. But in an absence of 5-methylcytosine or plenty of cytosines, the gene expression is switched on. Next slide, please. So we can imagine that ourselves, like we have a very, very active gene without binding through methyl groups, and when methyl groups are present, the gene is very silent, cannot express fully itself. And next slide, please. Next one, thank you. So in the presence of methyl groups, simply transcription factors cannot bind to methylated promoter and gene expression is blocked. In the absence, the gene expression can freely go on. And now, important thing is that that's a normal process, the steering through DNA methylation that happens in all healthy cells in proper time, proper place, and in proper amount. The problem starts when DNA methylation process is somehow disturbed. And next slide, please. Next slide one. Thank you. And on this epigenetic mechanism, also on DNA methylation, there's an influence of course, genetic factors. So that's somehow intrinsically regulated, but also metabolic factors, and we have a lot of disturbances from this side. And last but not least, and I would say most important, is environmental various environmental factors. Either it's just environment we live, or these are disturbing, damaging factors that are in our surrounding, and all those have an influence and change epigenetic mechanism, and that has implications on chromatin structure and, of course, gene expression. And next slide, please. Next slide. Thank you. Therefore, when we speak about health and disease and balance in between them, of course, we still want to be more healthy than disease. We have to think not only about genome, which we are very, very familiar on, but also epigenome with all these mechanisms steering gene expression. But all of this, they work together and they produce actually exposome, or exposome is the thing that takes everything from genome and epigenome, but adds this, let's say, local or environmental factors with the regards of toxic compounds, radiation, drugs, and of course diet, smoking, everything, what can imply our methylation. And next slide, please. So we would like to actually, if we think about markers and the measuring of any effect, we would like to know the healthy state. And what's the healthy state of DNA methylation? Of course, you can measure that with various instruments and various methods. And the methods I would like to share with you today is very simply because we just measure the total amount of M5C in relation to other cytosines. And the R is actually our factor that expresses total DNA methylation. And what we can see here is the methylation in generally healthy people, that was our control cohort, is around 2. Of course, it goes a little bit down with age. However, still, it's quite, let's say, high. But as you can see, the first observation is with age, there's something happening with DNA methylation. And the next slide, please. Next slide. And you should see our results for cell cultures. So the first bar are young cells, then middle-aged cells, and then senescent cells. And you can see the methylation goes down. So it goes down with normal processes. With senescence, with aging. However, next slide, please. We observed a lot of diminishing of DNA methylation in various diseases. And I would just show you some examples. And this is the example of total DNA methylation measure for hypertension patients. And you can see for control group it's quite high, but for mild, moderate, and then severe hypotension, it goes slowly down. Next slide, please. The same was observed for other common disease like hypertriglyceridemia, also with higher triglycerides. It goes down. And the next one, please. Next one, you see that's such a simple reaction, the organismal reaction to environment as pain. So we can have acute pain and chronic pain. You can see here the problems of the patients with, or the methylation of the patients with more acute, more short time pain, like up to 6 months and over 6 months. And we see the more pain and the longer pain we suffer, the lower methylation is. And the very, I would say, picturing. Next slide, please. The experiment we made. These are brain tumor tissues, actually quite benign brain tumor, which is meningioma. And these are 4 different tumors, and each of them, after sampling, was divided into 3 pieces. One of them, which are violet bars, were freshly frozen. Then the green bars were just left for a few hours in the normal room temperature and conditions. And then the third bar, which is yellow bar, was normally treated as pathologists do with formalin and paraffin, and then of course cut into pieces. So you can see with these environmental factors, the DNA methylation goes down. And next slide, please. Next slide, please. And here you can see the summary of actually our experiments and our measures for total DNA methylation for different pathologies. So you can see that global hypomethylation correlates well with severity, let's say, of disease and with more malignant diseases in case of neoplasm. So how can we use that, or can we look through this DNA methylation when we think about COVID and SARS infection? Next slide, please. These are like sample 3 slides now, showing you the recent from this year or from the end of the 2020 nice publications showing how DNA methylation changes during SARS infection. And here the authors show that host methylation can predict you depends on methylation score, as you can see, how whether the patient will be infected or rather not. The next slide, please. Next slide. On this, in this paper, the authors detected different signatures depending on severity, DNA methylation on severity of disease. And in the next slide, you see the paper that shows you that DNA blood methylation is quite important, or shows very big differences in case of the outcome of the disease. That's the next slide, please. And there were different findings in that paper. A lot of genes were hypermethylated in the promoter region, but also in most of the patients, 75% of acute illness associated DMR, which were located near promoter regions, they were hypomethylated. So we can see that general damage of methylation is quite important factor connected with severity of disease. And next slide, please. And we all deal with pandemic. We all know actually this picture or some other similar pictures showing the coronavirus with the spike protein, which is quite important for its pathological functions. Next slide, please. The coronavirus, the SARS virus, is RNA virus, and it enters the cell, the pneumocytes usually, through receptor angiotensin converting enzyme receptor 2. That's the main point of entrance, using spike protein. And so why is the pneumocytes, so respiratory region, important or so influenced by the disease? It's just because it has a lot of that receptor. So it enters the cells, and inside the cells it fuses with the membranes, and so we have virus release. Next slide, please. And when, of course, the virus is present in the host, so in humans mostly now. So it activates the whole inflammatory cascade. We observe lymphopenia, lymphocyte activation, of course, with all the markers, and cytokine production, cytokine storm. We see the disturbances in granulocytes, monocytes, and of course complement activation. So the whole big cascade is activated. However, what is then? What has that in common with DNA methylation? Probably something. But next slide, please. Recently, there was a paper published. You see the title and authors above. And these are two graphs from that paper, which shows you the differences on the D slide in neutrophil count between control patients, so healthy patients, those patients who survived the disease, the COVID-19, SEV-R, and those who deceased, SEV-D. So you can see in the first panel D that neutrophil count was generally lower in healthy patients than those diseased one, and also with those who survived were lower than those who didn't survive. Okay. And on an E panel, you see the ROS reactive oxygen species count in those patients. So you also see that reactive oxygen species connected with this neutrophilia is lower in healthy patients, and of course lower with those who survived than those who deceased. Next slide, please. Of course, reactive oxygen species are present in our organism all the time. The problem is, of course, the balance. So when our organism is healthy and in balance, it can manage some excess of ROS. But if we have overload of reactive oxygen species, simply the organism cannot do that anymore, and of course, it will go further with the damage. Next slide, please. And when we have reactive oxygen species. The most potent of them is hydroxyl radical. You see on the picture, they can react very fast, widely with anything they meet. It's not specific. They can react with lipids, proteins, sugars, and also nucleic acids, and then produce, in that case, modified bases. So that's one thing, that the virus produces increase of reactive oxygen species. On the next slide, you see the other, next slide please, the other problem that arises during SARS infection, when we see the binding with AC receptor and then entrance to the cell, and then of course out of cell ways of releasing more iron. That, of course, can cause DNA and mitochondrial damage. We call it something like ferrosenescence. And that can lead then to, of course, critical illness in COVID-19 and neurodegeneration. Next slide, please. Here you see the more focus on the damaging activity of iron through, of course, activation through SARS virus that goes directly to DNA damage, and that produces, of course, quite easily some neurodegenerative disease and aging, of course, of those cells. Next slide, please. Next slide, please. And of course, we can see the increase of iron inside the cells and outside the cells. What you can see on the slide, and last slide shows you potential ways to overcome this excess of iron. Can you see? Can we see the next slide? When you see, I hope. Oh, okay. So we don't see the next slide, but can you leave that? Can you leave that on the? You can leave that, please. Leave that. So I will just. Of course, you can get rid of iron when you use some iron chelators. That's, of course, the thing you can do. But what is the problem with excess of iron in the context of DNA methylation and oxidative stress? The iron is very, very potent cofactor of Fenton reaction, in which hydroxyl radical is produced. So, in one side, more more reactive oxygen species because of the cytokine storm and neutrophil release, and then more iron, and then more oxidative stress. And what that hydroxyl radical does, of course, it modifies bases. And what we can see is it modifies 5-cytosine, so it damages that. It can produce many hydroxymethyl cytosine, one of them. But it produces many modified bases, but also modifies other bases like guanine, and produces 8-hydroxyguanosine, which is a quite important and very clear marker of oxidative stress damage. The next slide, please. On the next slide, you see the table of the diseases in which increased levels of 8-oxoguanosine was confirmed. And of course, you see like aging, infections in the first place, we should say, and also cancer. So please, the next slide. And we just come to the CNS, to central nervous system, the brain. So really the main center of our nervous system is very vulnerable to oxidative damage, because it highly utilizes oxygen, of course, for its function. So it also, because of the structure of the cells, it has increased levels of polyunsaturated fatty acids, really for attack of free radicals. It has high level of redox transition metal ions and low level of antioxidants. So if we have iron in an oxygen-rich environment, that of course increases the level of hydroxyl radicals and leads to a cascade of oxidative events. Next slide, please. what we made before, of course, pandemic, we analyzed those two markers, 5-methylcytosine and 8-oxoguanosine, in brain tumors, so very, very malignant diseases of the brain. And on the upper level, you see the 8-oxoguanosine levels in different types of malignant gliomas. So 1 is very benign tumor, and 4 is very, very malignant tumor, glioblastoma. So you can see the steady, or well, I would say logarithmic increase of 8-oxoguanosine and with the malignancy or increase of disease. And on the bottom panel, you see the contents of M5C, and you see with increasing malignancy of disease, it goes logarithmically down. So next slide, please. Next slide, please. In this slide, you see very, very clearly how those two values are combined together, or they are related to each other. So once again, the more malignant the tumor is, shown that here in red spots, the more 8-oxoguanosine it contains and the less 5-methylcytosine it has in its DNA. So we clearly see the brain pathology, the malignancy of brain pathology is regulated, or it depends on oxidative stress, and also this effect, it's influencing epigenetic mechanism, and especially DNA methylation. And of course, the answer, what will come after pandemic, especially in case of CNS disturbances, is of course unanswered, because we are still in the pandemic time, and these long-term consequences are simply not known. However, knowing all the bases that I showed you, we can actually predict very, very set thing, that probably the level of neurodegenerative diseases and malignant diseases in the brain will unfortunately increase, because that's how the mechanism works of the disease, and of course the special effect of the disease to the brain. Next slide, please. Next slide. You see here one example. There are many published about what is seen on MRI pictures of the patients who survived the COVID. So of course, during the severe disease, they can have many pathologies like ischemic infarcts or also bleeding, and of course some clots in the vessels. That can all be, and of course these are acute situations and it has to be treated. But the patients who just survived COVID, and they don't really have big neurological problems during the disease. They are some of them are controlled, or they have the MRI pictures after, and what we can observe is something I hope is seen here. These are the white spots that you can see in this. I don't know whether my cursor is showing, but on the left upper, you see very small spots, many of them, and on the upper right also, and lower left the same. And these are flare hyperintensities. This kind of picture is connected with many diseases, many ischemic diseases, or the primary stage of the brain tumors. So we can clearly see that already in some well tolerated disease, the pathologies in the brain happened, and well, what comes from them is of course the question. So can we, knowing all the pathomechanism of the diseases and the pathomechanism of possible consequences, can we do something to protect ourselves from those long-term disturbances. And can I have the next slide, please? The next slide, please, of course, is we have to act on the opposite way. So if we have big oxidative stress, and that's the most potent factor that influences a genetic mechanism, then we should act through possible antioxidants, let's say in a very general way. And as you can see here in the slide, a lot of possible substances is named, the natural substances, but they are probably the best. I just remind you that still, with all these clinical trials, we don't have a nice potent drug against SARS, also no really very effective vaccine. Next slide, please. And I would like to show you the most potent antioxidant ever, which is lycopene. That's its structure. And why is it so potent? Because of this many double bonds that can catch reactive oxygen species and hydroxyl radicals, especially. Next slide, please. And this lycopene is a red dye of tomatoes and some other foods. However, in tomatoes, especially ketchup or sauce, those have the most content of that. So manufactured tomatoes are the best product, and of course you can have a special drugs containing a lot of lycopene. Next slide, please. I have just two more, and you see why in this upper panel, lycopene has most potent singlet oxygen quenching compared with other carotenoids, other antioxidants. And the last slide, lycopene already showed nice action towards many neoplastic diseases. Can we see that on the last slide? They have thank you very much. Those are as a summary, the already known positive reactions of lycopene working through this antioxidant mechanism, quenching the hydroxyl radicals and other reactive oxygen species, and that's the way why it stops or slows a neoplastic process. So through the parallel thinking about reactive oxygen species action towards DNA methylation and its implication to epigenetic, of course, we can that can support from one side the mechanism of action of pathological action of SARS virus towards epigenetics, but also shows you the way how to overcome the disease. Thank you very much for your attention.